I'm Meredith Blackwell, and I'm at the International Mycological Congress number 11 in San Juan, Puerto Rico at the Convention Center. And I'm going to talk here to Nina, who will introduce herself.
So I'm Nina Gunde-Cimerman. I come from Slovenia, from Ljubljana, actually from the University of Ljubljana. And I'm also going to talk about fungi, I guess.
Good. Yes. And just as an aside, my son-in-law is Slovenian.
Oh, really? How that did
Several
happen?
generations.
Okay.
The name is B-A-J-O. Do you know that name?
B-A-J-O.
The name been changed.
Oh, yeah?
Bejo, they call it.
Okay.
Yeah.
But that's actually quite difficult because Slovenians, we are only 2 million, so
Yeah.
you are actually in a very exquisite club to get somebody Slovenian in the family.
Mm-hmm. So my granddaughter is half Slovenian.
Okay.
So why don't you tell me about where you grew up? It was always in Slovenia that you lived?
Well, I was born in Ljubljana, I was born in Slovenia, but when I was 8 years old, my parents decided to go to Libya, out of all places.
Oh.
And I was going to an Italian school for 4 years. Nobody asked me if I wanted or not, they just put me in this school. So first it was Italian school, then for a short while it was an American school because, the revolution happened, Gaddafi came, then we went back to Slovenia. So I had a bit of a childhood, I can say.
Yeah.
And after that...
How old were you when you went back?
When I went back,
Slovenia?
something like 13 years. And then I slowly entered high school in Ljubljana, went to biology. So I studied biology, and at the same time I wanted to study architecture, languages, philosophy. So it was totally a matter of coincidence that I ended up at biology. Not one of those that knew from the cradle on what I would want to do. So then when I finished biology, I had to do my undergraduate thesis, and I got a position at the National Institute of Chemistry at that time. And actually for studying gamma mutations in Aspergillus niger to get a better production of cellulases. It was a project that was at that time financed by International Atomic Energy Agency, and I thought it was very fancy. Anyway, I was really happy because based on these results that I obtained, we published an article which was recognized, and it was only my undergraduate thesis, so
Wow.
I was really happy. Somehow, because of that, when I finished my school, when I finished university, they offered me a position there, so to go and have a master degree. So I did have a master degree at the National Institute of Chemistry, and at that time, we were in Yugoslavia. Slovenia at that time was part of Yugoslavia. A socialistic country, not that closed as, for example, Eastern Europe and so on, but mainly not closed in the sense that you couldn't travel, but closed particularly because we didn't have much money. So at that time there was an opportunity, and the opportunity was that one could apply for a USDA scholarship. So it was given by the research center, the USDA Research Center in New Orleans. And for the entire Yugoslavia, which was 22 million people, they actually offered 2 positions.
Wow.
I did
Both
apply.
in New Orleans?
Yeah, the one in New Orleans. I did apply, and I was extremely lucky because what they looked for, and I didn't know that in advance, was actually that somebody would really have quite a reasonable knowledge of English, which at that time in Yugoslavia was not that common. But since I lived in Libya and had some experience with the American school, I did really quite well. Not because I was smart or anything, I just spoke a little bit better.
So luck.
Yeah, so one of these 2 scholarships went to me, and I ended up in New Orleans, and I did my master degree there.
And
Well, I defended it at home in Ljubljana, but I did the experimental part there. Yeah.
So it was through the university.
Yeah, so I defended it there, and I had a totally wonderful time. You can imagine a student from
Did you go to the
Yugoslavia.
French Quarter much?
If I did what?
French Quarter. Did
Yeah,
you go to the
of course
French
I did. I went to the French Quarter, I went to listen to jazz, so I was like 10 hours per day in the lab, and then time off. So I slept very little, in short. That was not something I did much.
It's one of my favorite cities.
It's a wonderful city, and I do remember it, of course, like it was before Katrina, because I haven't been after Katrina happened. So I have really these beautiful memories of New Orleans.
But I think they're bringing it back as they did here.
Yeah.
After hurricanes, they try to get it working fast. Yeah.
So it would be nice to go. I would like to go,
Yeah.
but yeah, there has to be an opportunity.
Yeah. Now, Slovenia is that where the Dalmatian coast was?
Yeah, Slovenia borders on one side to Italy, then to Hungary, and then to Croatia. And Croatia has the Dalmatian
Oh,
coast.
it's Croatia.
Yeah.
That does. Yes, I had a
But
very good friend who was Slovak.
Yeah, Slovenians, we traditionally go for our summer holidays, we go to Dalmatia. So this is what we do. We go to their islands, they're really beautiful. So yeah, we also did a lot of that.
Yeah, I had a third friend. He painted pictures of some of the cities on the Dalmatian coast. So,
Yeah.
yeah. So now what do you do?
Well, maybe I just should say that then I made
Oh, yeah, you didn't get
my
your PhD.
PhD.
You have to do
So
that
I
part.
did my PhD also at the National Institute of Chemistry. And at that time the theme was finding a replacement organism and fungus for the statins which were patented and being produced by Aspergillus terreus.
Right.
So Merck had a patent on that, and this Yugoslav-Slovenian pharmaceutical company wanted to find another producer. At that time, once going through the market, the food market in Ljubljana, I saw all these mushrooms being sold, and I thought, why wouldn't one look in the Chinese pharmacopoeia? Because as in Europe we have this tradition of these herbs that have medical effects. We all know that Chinese, Japanese have this kind of tradition. So I got somewhere some kind of pharmacopoeia and I studied it, and I discovered that for Pleurotus ostreatus, the oyster mushroom, it was actually written that it has this effect, anti-cholesterolemic and so on. So, well, anyway, my PhD was then to prove that oyster mushrooms and other Pleurotus, not only the ostreatus, do produce all of them statins. And it's a very good source of, lowering your cholesterol. You just dry the mushroom, put 1 spoon of something in your yogurt, of it in your yogurt. It's much better than taking the pills.
So you can actually eat it?
You can actually eat it and you get the same effect, and it's a kind of synergistic effect. So of course the PhD was not that simple, but we identified the substance and did the fermentation and discovered how it moves from the mushroom into the spores and all sort of things, and it was nicely published, and the company made a patent on that. So that was my PhD.
Yeah, I've actually heard there are a number of fungi that produce statins, some of which are commercial now.
Yeah.
But Terreus is a fungus I once found in hay. It was all over the hay, damp hay, that a horse had been fed.
Yeah.
And the horse died. So, too much of a good thing.
Yeah.
They're
Well,
other toxins.
Terreus is not what you want, but oyster mushrooms are quite nice.
Yeah, that might be
But
a good thing.
via the oyster mushroom, we actually come to what became my subject when it comes to my research. Because I told you that this Slovenian pharmaceutical company made a patent, an international patent. But after they made this patent, nothing happened. All sort of people approached us, whether shouldn't be open a spin-off and things like that. But the pharmaceutical company was like, they didn't want to say anything. And eventually I discovered that what they wanted with this PhD of mine was to have a kind of smoke screen. In reality, they wanted to produce by the technology which was owned by Merck. So they just needed something to wave so nobody would pay attention to what they in reality they were doing. And since I was young and idealistic and took things very seriously, I was so disappointed that I decided, okay, now I want to have a theme that is going to be totally mine and totally new and totally away from all this kind of industrial interests. And at that time, I was aware, it was a fashion in microbiology, I can say, that bacteriologists are crazily finding all sort of interesting bacteria and archaea in extreme environments. And I thought, okay, which extreme environment do we have in Slovenia? And realized that we have these beautiful salterans which originate from Roman times, so they're 2000 years old. And they're also known that in the 16th century, microbial mat has been introduced from Dalmatia that you mentioned before, from an island in these salterans. And because of that, what the soil they produce in these salterans is totally white and pure, because no mixing occurs from the mud beneath and the crystallization of salt up. And then the next thought was, is anybody doing fungi in extreme environments? Does anybody has ever looked in fungi in the salterans? And it turned out that the only knowledge we have at that time, that was around 2000, is that fungi can contaminate food preserved with lots of salt, but absolutely nothing from nature. So I started working in these salterans. And to cut the story short, not to make this interview too long, we did indeed discover that certain groups of fungi do grow extremely happily in the salterans. First we found them in Slovenia, then on a global scale, and that they're adapted to the salterans on all sorts of molecular levels that we have been studying ever since. So my career of mycologist specialized on extreme fungi started due to a disappointment that was turned in a good way in the local salterans. Yeah.
So tell me which fungi in particular are in the salterans.
So in particular, the fungi that do best are the black yeasts, and there's one which is all over the world the dominant one, and that it belongs to Capnodiales. It's called Hortia werneckii, and that's the only organism, bacteria included, that can grow throughout the entire salinity range, which means without salt and up to 32% of salt, which is precipitation level. No, seawater has 2 to 3%. Here we are talking 10 to 12 times more.
Okay, percent. I was thinking parts per thousand.
So this one is, like I said, no bacteria is known to be able to that, 0 to 32, and it has a very broad optimum, around 5 to 10, and it's a perfect model for studying molecular adaptations. So we've been studying its population dynamics, genomics, molecular adaptations, biotechnological potential, enzymes, name it. So this is the main one, but it's not the only one. Then you also have other black yeasts, which are less frequent, but still, like Trimatostroma, Phaeotheca. And there's quite a lot of Cladosporia, so the number of them. Maybe you know the Cladosporium halotolerans is one of the most known Cladosporium, so that one was first discovered in the salterans, so it comes from there. Now it's all around the world. And then also some white and red yeasts, like for example Rhodotorula is also present. And then the interesting thing for me is that you don't find marine fungi in the salterans. For marine, this is absolutely too extreme. These fungi that I said are really specialized for these hypersaline environments. And then, well, the story continues to another extreme environment. And this other extreme environment that we are studying is the Arctic glaciers. Because, while working with these halophilic fungi, we made a realization that it's all about low water activity. So if there's a lot of salt, it binds water. So the organisms live in an environment which is low in water. And when you have ice in the glaciers, well, water freezes. And when it freezes, it lowers the water activity. But at the same time, it also creates channels around individual ice crystals, which are full of brine, which are actually brine channels. So it's a very similar environment to the salterans after you think about it. It was not known. So in some years after the discovery of this fungi in the salterans, we very bravely, you still have to have it in mind that I come from the part of the world where maybe it's less privileged in that way, applied for a European project which allowed you to do work in Arctic, in Svalbard, based on this hypothesis that I just explained. Nobody has done it before. And we got it. And I went to Svalbard. I was extremely happy. I was the first Slovenian ever. Woman, man, it didn't matter.
A woman Slovenian.
Yeah, I was totally happy. But the main idea at that time was to look for the fungi in sea ice. But global warming was already plague, I can say, because that time when I came, there was no sea ice. It simply was already melted.
What month was it?
Yeah, that was May, and it was already gone. And I was so frustrated because I had these 3 weeks and what to do, what I'm supposed to do. That out of frustration, I went, there was a very nice British guy in this station who was kind of fun. And out of frustration, to release my frustration, he helped me. And we went with the boat and we collected pieces of glacial ice that were floating in the sea. And then I started to melt this glacial ice in the lab. And to my huge surprise, it was full of algae. I mean, full. You know, we are talking 10 to the 5th per milliliter.
All dark.
No, mainly Cryptococcus, Rhodotorula, but if you had ice with gypsum inclusions, which is something that you also find in the salterans, then it was all black. Well, first, you know, the blind chicken syndrome. Huh? So first you get the results, then you find the hypothesis, was that kind of thing. So we had, how is that possible, really? How could that happen? Anyway, after quite some time, we realized that we probably, by chance, discovered the populations which are formed in the so-called subglacial environment. So if you have a glacier, you have the ground below, the permafrost below, and then you have this mass of ice on top of it. And when this mass is heavy enough, it presses on this ground and creates a very thin biofilm of water. So whatever falls like snow on top of the glacier then travels layer by layer by layer by layer until it reaches the bottom where the liquid water is, and whatever survived gets enriched there. So this passage can be 50,000 years. But then if it survives, then these populations of yeasts are created, I can say it like that.
Yeah.
So then after we got this hypothesis, we systematically sampled and so on, and now we know this is like that. So we are sure about this, and this is our second group of organisms that we are studying. And of course, it's a very interesting phenomenon because with this global warming, these big pieces of glaciers are, you know, falling off and melting, and all these biomasses go into the oceans and they travel around the world in the bottom of the oceans. I mean, all this nitrogen on one hand and also all this diversity that nobody really knew before. So it's quite a phenomenon. It's a very interesting system to study, and we go on doing it.
Are they different, distinctive species?
They are distinctive species. We have a very, the diversity is quite high, but the dominant species are not that many. So it's, like I said, several species of Cryptococcus, Rhodosporidium, Rhodotorula, black yeasts, Aurobasidium in particular, and some more. These are the core group. And then you have a lot of this, you know, kind of small sporadic variety that is interesting, but not from the perspective we want, because we like to study the molecular mechanisms. So we studied a lot about the cold adaptation, like on the fluidity of membranes, on the level of compatible solutes, cell wall, polysaccharides which are formed, all these things. And so this is our second big environment. And then comes a third one.
Another extreme environment.
Another extreme environment which is totally different. And again, it comes in a way from a frustration. I guess this is a kind of repeating motive.
Frustration leads to discovery.
And in this case, I was at home, sick, the flu. And when I got well enough, you know, to not have to stay in bed, I kind of could circulate around the apartment and be impatient. I once stood in front of my dishwasher at home. And I looked at this dishwasher and said, this is a domestic extreme environment. It has either lots of water or nothing. It has pH up to 12 due to detergents, oxidative stress, and temperatures, you know, 60, 70 degrees. So this is really an extreme environment. So I sampled, brought it to the lab, and 1 week later I got my assistant, my colleague, came to me completely white in her face, and they said, Nina, you know what you have in your dishwasher? I said, no. I mean, I'm not a perfect housewife, but, well, what do I have? And she told me, I have like a huge amount of a black yeast, again black yeast, Exophyllis dermatitidis. Exophyllis dermatitidis is a very nasty human opportunistic pathogen that we don't know actually anything about it when it comes to its natural sources. I mean, there are few isolates in the entire world coming from some berries in the tropics, but maybe 10 isolates.
When were dishwashers invented?
Yeah, and it looks like all the other isolates that existed at that time came from patients. And this Exophyllis dermatitidis is very frequent with cystic fibrosis patients. 20% of them in Sweden have it. It goes into the brain. It creates cutaneous, subcutaneous infections, lungs, all sort of things.
Were you able to do a study to see if it was mostly women or people who had dishwashers?
No, it was not that simple. Anyway, so wanted to say that then I really got worried, and then we looked a little bit, and we saw that in the other literature, they only associated Exophyllis so far with a little bit with sinks in bathrooms, a little bit with showers, such things, but not high numbers. We had 10 to the 6 cells per square centimeter, so of the rubber seal where we isolated. Okay, so like I said, I know I'm not perfect, far from that, so I asked my colleagues to do the same sampling, and all the dishwashers had the same results. Then we asked students, because they work at the university, and they brought samples from all Slovenia. 70% of dishwashers were fungal, and 50% of all isolates were this Exophyllis dermatitidis. Then followed colleagues around the world, so we got many samples, and to make it sure, 50-60% of all dishwashers globally are infected. And the interesting thing is, it doesn't matter if you sample in the Arctic, in the tropic, everywhere you have the same 5 species. And the dominant one is Exophyllis dermatitidis. The other 4 are also all opportunistic pathogens. So it seems the selective pressure in dishwasher is so strong that regarding which water, which food, which person, you get the same fungi. Yeah. So this was one of these stories that happen sometimes that were totally viral. So Yellow Press took it and yeah.
Well, let me tell you about that. 3 years ago, I was on a fellowship at Harvard, and I had to give a talk. And so my talk was about fungi everywhere, and fungi for all the people that were in the program, because many of them weren't even biologists. But everybody practically had a dishwasher.
Yeah.
And at the same time, there was this horrible outbreak of all sorts of meningitis caused by your organism. And in Boston, there was a pharmaceutical company there.
Yeah.
You remember this?
Yeah, I remember that, yeah.
And so anyway, I was talking about that because the trial was going on at the time. And then I was able to show the picture of the dishwasher. So it worked out because of you.
Okay, I'm glad I contributed something.
Excuse me.
But when you asked me whether we could prove that, for example, people that are using dishwashers have these infections, no, because it was extremely hard to come to any agreement with medical doctors, I must say. They are a bit of a hard population to talk to, was my kind of conclusion. So I kind of left this part, you can't do everything. But then we mainly focused on seeing how from the dishwashers, this Exophyllis dermatitidis gets spread into the environment. And we learned many things. So now we know, for example, that kitchens with dishwashers have a totally different microbiome than kitchens without dishwashers. So if you have dishwashers, you have high diversity of fungi, and you have these black yeast everywhere. I mean, on all the surfaces. If you don't have a dishwasher, I'm not saying that that's much better, but then you have Candida parapsilosis everywhere. So
I've always heard it's better to have a dishwasher and use high temperature to scald the dishes. at the
Exactly, yeah.
end.
I use it, but I just use it at the highest temperature. I don't follow much these ecological suggestions because if you use what they tell us, degrees, 40 degrees, which is very ecological, and we all know our body temperature 37, I mean, you actually, each time you press that button, you make a selection for pathogens. So you enrich them in your environment. And the same happens in the washing machines, by the way. We also study that, so the same thing.
So, has this given you any ideas about where to look in nature?
Yeah, well, the next thing, the newest thing we are doing, oh, you mean for Exophyllis? Now, we thought a lot, but the places we had in mind, so far we didn't yet find it. No,
It may be that there's so many organisms in those habitats, they compete, and
it's very slow growing under these kind of conditions, but we did find it in Arctic glaciers, to my surprise.
Wow.
Yeah, so it seems it actually can fall with the snow on the glaciers. And we also saw it in the water coming from the glaciers, and it's also in tap water, by the way, but in small numbers. But when it gets into dishwasher, it gets enriched. But what would be the natural reservoir? These numbers that we got around are too little that I could say that we know. In that way, we don't know yet. Well, maybe only one of those really kind of domesticated fungi. It's, there are quite some after all.
Yeah, what's its closest relatives? Or
Well,
that's been worked out, I'm sure. Somebody's done the phylogeny.
Yes. Exophyllis, they also belong to this Captodiales. So they belong to the Chaetotheriales. So they are actually related to rock-inhabiting fungi. So that seems to be the closest. Yeah. But there are quite many Exophyllis. They are known for their ability to be pathogens also on fish, for example, on amphibians. They are known for its ability to degrade aromatic hydrocarbons, so it's an interesting group of fungi that,
So they're not all high temperature?
No, they're not. You have the high temperature ones and the cold temperature ones,
Interesting. Very neat.
Yeah. So then the next thing, the last thing that we are very excited about now.
Number 5.
Sorry?
Number 5.
Is, again, throughout my professional career, I was in a way competing a little bit with bacteriologists because they think they have the monopoly when it comes to extremes, like fungi can do it. In this case, it's the same. We are talking about Greenland, which is becoming more and more black. Southwest Greenland is black because of this bloom of black ice algae. It's something that we know only for the last 5 years. And due to this ice algae, which have a particular pigment, purpurogallin it's called, that covers the chlorophyll, the ice is getting melted much, much faster than if it would be nice and white. So it's really accelerated the melting. And all this water now, of course, goes into these crevices, goes to the bottom, to the permafrost, and stimulates the methanogenic action of the archaea. So more methane is being produced, which generates more melting. So it's really totally accelerated. And with this phenomenon, now European project is dealing, which is called Black and Blue. It's led by a British group. You heard?
I've heard it.
Yeah, and they didn't have in their consortium nobody to look into the fungal part. Only chemists, bacteriologists, that was it. And somehow I kind of managed to convince them that it would be a good idea to look for the fungal part because they were sure there's nothing.
Yeah.
So only last year, in 2017, we were able to send our student, my student, Italian, very nice Italian girl, so she could join the expedition and go with them, and she sampled for the first time for fungi. And in short, there are absolutely really many.
More important than anything else.
Well, there are really, really many, only from this black ice, because we sample black ice, clear ice, supraglacial water, and these cryoconite holes. Only from here we got already 35 genera, not species. So genera, and we did the cultivation and also the NGS, next generation sequencing analysis. And since this was sampled only last year, we are of course working on that. So there's plenty to work on. These are the first results now coming up. But what I was really so surprised was that I saw there's a number of pathogenic fungi there and a number of endophytic fungi that nobody thought would be associated with this algae. I should also say that experimentation with this algae is very difficult because they're not cultivable at the moment. Nobody knows how to cultivate them. So everything you have to do is fresh biomass, which then dies off.
Yeah.
And then you have to go back again, if you're lucky, which is not that cheap, and get new biomass. So this is a totally new world of extremophiles that we are just discovering, and I guess in about 1 year I'll be able to say something more meaningful from what I just said.
Well, it's pretty exciting so far. No, that is really interesting.
Yeah, this is really what we are really excited about. And just maybe to make a little connection to the extreme domestic environment, it's quite funny. Another appliance that we are looking is refrigerators. We haven't published that yet. But when we look in the refrigerators, not anything related to food, just the plastic part and where you have this condensation, you know. What we find there is in 35% of the cases, it's Arctic fungi. Fungi that so far have been found only in the Arctic and not elsewhere. So they have a home away from home in our refrigerators.
So do they get from north to south and south to north?
Talking about endemism, yeah, well, this is also, we are doing some initial work on this level because we are doing also some genomics. So we are comparing Arctic and Antarctic strains of the same species.
With refrigerator stepping stones.
Exactly, yeah. So we are starting now this, and we will see if we have, if they're different, if they're the same, if it's just kind of ability to be dynamic. Remains to be seen.
I guess some of them could be carried in the
air. Actually little is known about that. It's also when you start working on this, you discover how little we know about this. How they get transported over these huge distances, because we're talking from one pole to the other.
Right.
So
there was I got my start in mycology because Dr. Exophyllis needed someone to look at an airborne fungus project he was working on. Not that I ever knew anything about it because I'd been studying fish, but I was supposed to look and see what I could grow out of slides that had been exposed to the air pretty high up.
Yeah.
Interesting work.
Yeah, well, it's something we should do more, altogether more of that, because, like I said, it would also help to explain some endemic questions. But on the other hand, as I mentioned before, now that we know that these glaciers are melting, and well, these cyclotolerant organisms then get released in the water, and if they travel with currents and so on the bottom of the oceans where the temperature is, you know, below 5 degrees, they can in fact reach the other pole without any major problem, I would say, if they're helped with the currents. So it's something which nobody, as far as I know, has looked into it, and it would be really interesting to do it.
Well, I like very much the interest in your house, and then it applies to nature too. It's very, very interesting.
Well, we like this combination. It's kind of, it puts you in another frame of thinking, so that you look around yourself in a different way, simply like your environment.
Well, this has been very interesting. Thank you so much for coming and being interviewed. It was your students who were pushing this, and I thought they wanted to be interviewed. No, not us, not us. And they went and got you. So I'm really glad that you came back.
So thank you for the invitation.